So my protein was the pyruvate kinase, which is found in the cytosol of the matrix, and it catalyzes the last step of glycosis for cellular respiration. And this is only in mammals that this happens, and that this pyruvate kinase protein exists. It has a quaternary structure and is found in 4 different places of the human body. Like the liver, most of the adult tissue, the heart, the brain, and fetal tissue as well. This is the ribbon model of the protein. It has an average of 500 amino acids with a different kind of secondary structure. Its primary structure is this, and it is a sequence of 20 different amino acids, and it changes depending on its location. So it's classified as a beta protein, but despite this, it has both alpha helixes and beta sheets, but the domain family that it belongs to is beta barrel. And like I said before, the quaternary structure has 4 different domains inside of the human: red blood cells, liver, muscles, and most adult tissues. It's a tetrameric protein, which means its quaternary structure has identical subunits that are dome to dome, and has dimer to dimer, which is a complex of 2 single proteins that are not bonded covalently. And by covalent bonds, I mean it's sharing electrons. The biggest issue that this kind of protein has is if it's not, if there's not enough of it, and because it catalyzes the last step of glycosis and it makes the output of 1 ATP in pyruvate, it pretty much funds the cell's energy. So when there's not enough and there's a deficiency, it can be almost life-threatening. And for red blood cells, not having enough of this type of protein doesn't have enough energy to transport oxygen throughout the entire body. So it starts to degrade as it's working. So, and the reason it doesn't is because inside the liver there is a mutation that challenges the gene that has the coding for pyruvate kinase, but it is not available. So that's this protein.